good afternoon, everyone. Welcome to the Yale Cancer Center Grand Rounds. Today we have two fantastic speakers, both who are in the Developmental Therapeutics program of the Yale Cancer Center. And why don't we get started? I'm delighted to introduce Dr. James Farrell, who is a professor of medicine and the director of the Yale Center for Pancreatic Diseases. He's an internationally recognized expert in pancreatic disease treatment and research, and is known for his development of personalized therapy approaches for pancreatic cancer and early detection biomarkers for pancreatic cancer. He received his medical degree from University College Dublin and completed internal medicine training at Johns Hopkins and a gastroenterology fellowship at MGH and Harvard Medical School. He then completed advanced therapeutic endoscopy fellowship training at Mass General and Brigham and Women's. In addition to his clinical practice in pancreatic disease and interventional endoscopy, his clinical research has focused on early detection of pancreatic cancer, including studying pancreatitis, high-risk individuals, and pancreatic cysts. His translational research includes the development of personalized therapy approaches for pancreatic cancer and the evaluation of biomarkers in pancreatic disease. Delighted to turn it over to you, Dr. Farrell, and look forward to hearing what you have to say. Thank you.
Great. Thanks, everyone, and good afternoon. Thanks for the invitation. So we're going to talk about pancreatic cancer early detection and prevention this morning. It really is going to be an overview in the time allotted and kind of emphasizing some of the work that we've been involved with, disclosures. So when you think about pancreatic cancer in terms of overall incidence, it's actually low down in the list and maybe sometimes doesn't get as much prominence as other cancers such as prostate or lung or colon. But when you look at cancer-related deaths, it jumps up the list and is probably around the third or fourth most common cause of cancer-related death. So in 2022, it's estimated there'll be about 62,000 cases and just under 50,000 deaths related to it, and has long been predicted and is certainly on its way. By 2030, it's expected to be the second most common cause for cancer-related death. It also needs to be emphasized the fact that, yes, there have been improvements in the management and treatment of pancreatic cancer, but really they have fallen short, although progress still continues to be made, and just emphasizing the need for a better and improved early detection for this particular disease if we're hoping to improve the overall survival. Now, there has been tremendous progress in the world of understanding cancer biology of pancreatic cancer. Along the bottom here is a normal progression through low-grade and high-grade dysplasia, or so-called Panc lesions, to invasive pancreatic ductal adenocarcinoma. It always typically starts with the activation of an oncogene, and then there's several well-known and characterized tumor suppressor gene mutations, including P53 and SMAD4. It's also worth noticing that related to this is another entity of the pancreas called IPMN, or intraductal papillary mucinous neoplasm. It also goes through a similar sequence, resulting in an invasive neoplasm with similar mutations such as in KRAS, but also some unique ones such as GNAS, a GNAS oncogene activation. But it's very hard to talk about carcinoma without talking about the other disease of IPMN. It's also worth making the point that this progression, we think, from the initial KRAS mutation to the time of, let's say, metastatic cancer, is probably of the region of about 11 years or so. And so this number is debatable, but basically it's a long time for us to try and intervene and understand what's going on during this sequence. However, it's also worth noting that in its early stages and pre-invasive stages with Panc, it typically can't be seen radiologically. When an early stage cancer does arise, this can be seen by radiologic imaging. But in the time required to go from an early stage resectable, surgically manageable pancreatic cancer to an advanced metastatic stage can be as short as 1 to 1.5 years. And so at that point, the clock does start ticking. And it brings up the issue of missed cancers, interval cancers, cancers that may be seen on imaging with more advanced uses of imaging. Now, there's no shortage of biomarker signatures for early pancreatic cancer. This is just a selection of some of these, and they all have very promising operating characteristics, or AUCs. So in the top left is a salivary-based 4-gene panel of RNA that we worked with, again, comparing non-cancers to patients with cancers with a very respectable AUC of 0.97. Below is work of Anup Sharma and Neeta Ahuja looking at DNA methylation markers, and this is a particular 4-gene cell-free DNA methylation panel, again, with very respectable AUC curves. We've kind of come to the age in 2022 where we now have the beginning of commercially available panels, not just for cancer in general. So you've heard of GRAIL and you've heard of CancerSeq, but now Immunovia has an IMMray PanCan-d, which is an antibody panel specifically for pancreatic cancer. And in the studies to date, at least, shows very promising AUCs for the differentiation between early stage cancer and control. And in fact, this is being currently tested in a high-risk population, the PanFAM-1 study, in which we're involved with, results of which will be out soon. But the problem is, yes, there are all these great biomarker signatures, but they're not being applied or can't be applied to the general population. And this has to do with an issue of mathematics and the 1.6 lifetime risk and the relatively low prevalence of pancreatic cancer. What that means is that even with a very good sensitivity and specificity, whereas you have a very high negative predictive value, you have a very low positive predictive value. And even as you increase the sensitivity and specificity of these tests in the general population, the positive predictive value only gets as good as about 20%. And what that means is that most of the positive tests that you're going to see in the general population will turn out to be false positive tests. And this is one of the major reasons why we haven't unraveled general population screening currently. So as a result of that, we tend to focus on high-risk groups. And the 3 big high-risk groups, which are these enriched groups that increase the prevalence of the chances of developing pancreatic cancer, include new onset diabetes. And I'm not going to go spend too much time on this today. Suresh Chari, who's kind of a leader in this area, spoke very passionately about this subject here at Yale several weeks back. But diabetes interplays with everything that we talk about with pancreatic cysts and family history, the other 2 high-risk groups. So what about pancreatic cysts? So pancreatic cysts, and specifically a type of cyst called an IPMN, are incredibly common findings, incidentally found typically on CT scans or MRIs of the abdomen. We use a variety of imaging features of these incidental cysts, such as high-risk stigmata. Are they associated with jaundice or a mass? Is the pancreatic duct, for example, dilated? Or worrisome features? Is there a nodule? Is there an intermediate ductal dilation of the main pancreatic duct? Is a cyst greater than 3 centimeters? Is there a change in caliber? Is a cyst getting rapidly bigger over time? So we use these imaging features to try and better understand what's the risk of this particular IPMN having cancer. And so we think that overall, there's a 1% chance per year of these IPMNs developing cancer. And when we look at the imaging, one of the first questions that we end up trying to sort out is, should these patients undergo surgery? And so we use a combination of the imaging, but even with our best imaging in 2022, it's likely that anywhere in the region of about 60% of patients are undergoing unnecessary surgery, meaning that we are resecting low-grade dysplastic lesions, not the high-grade dysplastic, not the invasive cancer patients that we're really trying to find. And so this kind of begs for an improvement in this particular area. And one particular area has been in looking at the role of cyst fluid. And so we have the ability with endoscopic ultrasound to pass a camera down into the stomach and sample the fluid in these cysts. And this can be very helpful for making the diagnosis of the cyst, like is it an IPMN or some other type of precancerous cyst. But it also has the ability for us to look at, for example, methylation markers. And this is very promising 2-panel marker that has a very good high operating characteristics for separating out low-grade dysplastic cysts, which we don't want to operate on, and high-grade dysplastic and cancer cysts. So I expect to hear more in time about these particular markers in cyst fluid, as well as other even protein-based markers in this particular field. The other issue with pancreatic cysts relates to surveillance. And so as we get better at deciding which patients shouldn't undergo surgery, it'll become obvious to people that the vast majority of these patients with IPMNs nothing really happens to them dramatically over time. They might get a little bit bigger, their duct might get a little bit bigger, but their chances of developing cancer requiring surgery is incredibly low. But we're obliged to follow them, especially younger patients, because we're telling them your cyst has a risk of developing cancer. And in that, the field of surveillance has really become a very complex issue, primarily because there's a lot of cysts involved. So it's estimated that about 6 million people in the United States at least have some form of pancreatic cyst. And it's brought up questions of, when should we stop surveying patients that have pancreatic cysts? Is there an age? Are there comorbidities that we should say outweigh the risk of cancer? Is there any way of tailoring or having kind of a frank discussion with patients to kind of educate them on who needs surveillance and who doesn't? And is there any progress we could make in the realm of understanding the biology of progression? So we know for a fact that cysts do change, some cysts do change over time, and some cysts will change even after periods of stability. On the flip side, we also know that stability, especially for small cysts, is a good hallmarker. It's not great, but it's a good hallmarker. Nonetheless, in a variety of guidelines that are out there to help us understand who we should be following, who we should be stopping surveillance on, the American Gastroenterology Association has made the recommendation that after 5 years of cyst stability, that you should stop surveillance. And this actually has been quite a controversial recommendation. Ankit Chota, one of the physicians in our research group, had the ability to look at about 18 studies with over 10,000 patients followed for almost 60,000 patient years. And Ankit looked at in these studies that have now long-term follow-up for patients with otherwise low-risk IPMN, so patients that we would typically just be following. And he was able to notice that, yeah, after 5 years or so, the risks of them developing progression is probably around 3% per year. And the risk of them developing a cancer or an advanced lesion is about 1% per year. And when he looked at the subgroup of patients, so patients who were stable for 5 years, so this would be the group that we would typically talk about stopping, there's still a 0.2% risk per year of developing cancer long-term out. So we don't think that there's enough evidence right now to really be talking about stopping surveillance. What we do know is that there are subgroups of patients who we should be probably having a more intelligent conversation with, and particularly patients who have significant comorbidities. We had the ability to look at 440 patients that we were actively surveying at Yale over a period of about 56 months or so. And in that group of patients, 44 patients died. But the vast majority of deaths on follow-up were not related to pancreatic disease. And in fact, when we took a cutoff of a comorbidity index of 4, so an index that takes in cardiac, renal, pulmonary issues to determine long-term comorbidity, a cutoff of 4 was very good at predicting long-term comorbidities not related to pancreatic cancer. And so now we're beginning to have these discussions with patients based on their overall prognosis, not just focusing specifically on the issue related to their pancreas. This very useful tool that's now available, we actually have it on our high-risk website, the PCD website, is a validated tool that was initiated and validated in both Dutch, Italian, and several US sites for looking at both the 5-year and the 3-year and 5-year risk of developing worrisome features of high-risk stigmata if you have a low-risk branch-duct IPMN. So you as a patient are able to go onto the website, you're able to put in the size of your cyst, for example. There's some other parameters like smoking, is your cyst multifocal, and it's able to give you a pictorial representation of your risk of the cyst developing into worrisome features and high-risk stigmata. So this is very helpful in discussions with patients overall. And ultimately, we would like to get greater numbers to be able to calculate individual risk of developing an advanced neoplasia. But I think a key issue is also trying to understand at a molecular level what makes some cysts stable for periods of time, what makes certain IPMNs decide that they want to take off after 5 years and develop into an invasive malignancy. And to this end, there is a need to understand IPMN cyst progression more. To date, there have been some limited data in the use of organoids derived from surgical resection specimens, so patients who've gone to the operating room and people have been able to develop IPMN organoids for additional study. We've taken a slightly different approach. We were actually interested in the groups of patients that we're surveying, so patients who do not go for surgery. And using, again, endoscopic ultrasound, we've been sampling the fluid from these pancreatic cysts and generating organoid structures. This is very preliminary data, and these are some of the images that we are identifying, similar to what we would see in organoids. To date, we have studied 9 patients with presumed IPMNs. They're all growing exceptionally slow, but we've been able to passage some of them as well. So this is an interesting development to allow us to study IPMN progression, to maybe tailor approaches for individuals, and maybe offer interventions such as chemoprevention. So stay tuned. Now, the other large area within the risk or the high risk of pancreatic cancer is your hereditary susceptibility or your inherited risk of developing cancer. Pancreatic cancer rather. We think that maybe about 10% of all pancreatic cancers are at risk for developing cancer based on a family history. And there's a variety of guidelines that we've been involved with, specifically the CAPS guidelines, to help us understand who we should be surveying and who we shouldn't be surveying. Very simply put, there's a high risk group that carries a very high lifetime risk of developing pancreatic cancer. It includes individuals with mutations in P16, Peutz-Jeghers, but also individuals who have a family history with 3 or 4 first-degree relatives. So these people are at a higher risk without a known genetic abnormality. There's also a low to moderate risk that includes genes that you're familiar with, such as BRCA1, BRCA2, other DNA repair genes like PALB2 and ATM. But also, again, in there is a family risk of 2 first-degree relatives that carries a significant, albeit lower risk than those that have 3 first-degree relatives, with no abnormality identifiable in the germline. To take care of these individuals, several years back, we set up a pancreatic cancer early detection clinic at Yale through Smilo, through the generous support of Smilo and Javier Lora's group. And in this group, currently, we're following about 185 patients, most of them in protocol, who are at high risk of developing pancreatic cancer. And again, you can see this from this group. The majority of patients are actually individuals who have a strong family history, but do not have any genetic abnormality. One of the original studies that was done in this area was what's called the CAPS-3 study. And these patients are typically imaged with a variety of CT scans, MRIs, and endoscopic ultrasounds. And a small percentage of them will have an abnormality that requires surgical resection. So in CAPS-3, for example, 225 patients were enrolled. A total of 216 were ultimately studied, and the majority of these at the time were familial patients. This is a multi-center study. For the majority of patients, no abnormality was identified, but you can see that for some of these patients had a dilated pancreatic duct, for example, a cyst, probably an IPMN, and a small percentage ended up having a solid lesion. In total, 5 of these patients underwent surgical resection. 2 of them had high-grade dysplasia, and 3 of them had low-grade dysplasia in IPMN, although all 5 of them ended up with having either an intermediate or high-grade dysplastic lesion in the form of a Panc lesion. Very interesting data from these CAPS studies, in fact, has been the ability to collect pancreatic juice. And what's been noticed in individuals who subsequently developed established pancreatic ductal adenocarcinoma is that investigators were able to go back and look at the pancreatic juice of these individuals at a time when they had no radiologic abnormality. And they were able to identify either combinations of P53 mutations or P16 mutations long before a radiologic abnormality was identified. So this is another direction to go in terms of surveying these patients. The initial results of the CAPS-5 study, which is a much larger multi-center study of which Yale was part, have just been accepted for publication in JCO. In CAPS-5, almost 1,500 patients were studied. A total of 9 screen-detected pancreatic cancers were identified, of which 8 were resectable and 7 at a stage 1A. And when you put that together with some of the high-grade dysplastic lesions, well over 50% of the patients enrolled in these studies had an achievement of what's called a successful goal of surveillance. So we identified either an early-stage cancer or a high-grade dysplastic lesion. When all the patients from the CAPS 1 through 5 study were amalgamated together, what we were able to demonstrate was that those patients who were diagnosed during a surveillance protocol with pancreatic cancer had a much higher median survival than those that were diagnosed outside a surveillance protocol, showing some form of survival benefit within this initiative. Anka Chodo, again, being very productive, went and looked at some of these surveillance studies that were done, more concerned about what was happening, not so much when patients were initially enrolled, but as they were followed over a period of time. He took 13 of the most recent studies, and what he noted was that in 53 patients that were identified with pancreatic ductal adenocarcinoma in these 13 odd studies or so, whereas 22 patients or 41% had goals of surveillance achieved, so an early cancer or high-grade dysplastic, a very worrisome, almost 60% of patients had a more advanced lesion, including metastatic lesions. He tried to look at some of the factors that might be associated with these late-stage presentations. It had nothing to do with surveillance modality, baseline imaging abnormality, even the presence or absence of a germline mutation. A lot of the patients who presented were actually asymptomatic. We were able to look at the timing of preceding imaging, and most patients had a normal preceding image. There were some questions about diagnostic errors in the limited data that was available, and there was some issue of surveillance adherence in some of this data. And what this leads into is kind of a growing conversation about how we can do better. You're going to see more and more of these types of studies in the next couple of years or so. This is the initial study that proved this point. These are patients who had a diagnosis of pancreatic ductal adenocarcinoma made. And when they went back over a year or so, it was said that a pancreatic cancer could be identified. So a variety of methods were looked at in these individuals, but it had to do with issues relating to abnormalities that were identified on imaging that could be seen, and maybe radiomics or some other feature could help figure that out. I think it's important to say, just to summarize, sorry, just a glass of water here. That the stage of pancreatic cancer at an early stage is improving, especially in younger populations, and the overall survival related to early stage pancreatic cancer is increased from about 44% in 2004, and now getting closer to about just over 80%. This is unclear why this is happening, but may be related to increased use of abdominal imaging and perhaps some of the surveillance programs. So we're beginning to gather some information about the benefit of both the surveillance programs as well as the outcomes related to those surveillance programs. And I'll leave you with this note as something to kind of look forward. Within the CAPS cohort of patients at Hopkins currently, there is now open a mutant KRAS targeted vaccine for patients at risk of developing pancreatic cancer. This is a phase 1 study that requires multiple intramuscular injections over a period of time. The initial outcome is for safety and tolerability, but they are going to look at changes in KRAS-specific CD8 and CD4 T cells at 2 and 4 years. I'm also interested to know that Moderna does have an mRNA-based KRAS target. These are typically being used in oncology trials, but may actually have a role in the high-risk population to ultimately prevent the development of pancreatic cancer. So in summary, therefore, just looking at the time, we currently are not performing screening in the general population. We are focusing on high-risk groups such as pancreatic cysts, although we're trying to get better at surgical selection, but also get better at tailoring individuals who should be surveyed and those that should not be. With respect to the familial pancreatic cancer cohort patients, we're getting better at documenting some of the outcomes associated with these studies, really with the view to improving those outcomes long-term. And I think you'll hear more about new onset diabetes. It certainly plays a role, and it helps us stratify our patients with pancreatic cyst and familial pancreatic cancer. And I would use this talk just to say that there are plenty of opportunities within our group for collaboration, for additional biomarker development and validation, but also in the realm of imaging. And just to say that really it takes a group of people to try and keep these studies going. This is a picture of Ankit, who's been incredibly productive and helpful getting our group up and running. There's several other individuals such as Scott and Aziz and Ling helping on the study side, as well as our really close and productive collaboration with Dr. Hooch and Dr. Sharma's lab. Thank you.
Wonderful, Dr. Farrell. Thank you so much for that really nice overview. I don't see any questions in the chat, but if there are those watching who have a question, please type it in and I'll make sure to ask it. I have a question for you. So I'm thinking about the sort of the root cause of the increased incidence in pancreatic cancer, because we're seeing an increase incidence nationally, but also in the state of Connecticut, correct?
Correct.
And so it's either, I can't remember what you said, the number of pancreatic cysts of the population that have pancreatic cysts. It seemed like a quite large number.
Typically 6 million is a number that's quoted. Yeah. If you would do extrapolation from imaging studies and extrapolate that to the general population.
So are we seeing an increase in the incidence of cysts, or is it diabetes? And therefore with diabetes, is it obesity? So what do you think is the root cause driving this increased incidence?
So there's different explanations given for the different increases, specifically for pancreatic ductal adenocarcinoma. It's felt that it's also related to an aging population. So as the population ages, that's driving up the overall incidence of it. So that's one explanation. But for sure, there are other issues with it. And so issues related to metabolic explanations and obesity would be one explanation. Trying to link that then to pancreatic cysts, the reason for the large volume of cysts, our perception that there are more cysts, is really to just do with the use of imaging. So the prevalent use of MRI scans, CT scans has really been driving why we see more cysts. Also, imaging scans are getting better, so we can see 3 millimeter cysts, 2 millimeter cysts on MRI scans done routinely. Now, the question is, can finding those cysts be converted to a better early detection strategy for IPMNs? Or for pancreatic cancer, the issue is that maybe those cysts account for 10, 15% of the total volume of pancreatic ductal adenocarcinoma.
Okay, thank you. David Rim had a question. Stage 1s went from 40-some to 80-some% survival. Is that due to greater diagnosis in stage 1, and have high stage diagnoses decreased?
So this phenomenon is from SEER data, and all the positive glowing phenomenon has been related to stage 1A. So it has been the increase in incidence, the increase in survival associated with stage 1As, the decrease in age of diagnosis, and it's not seen in the other stages. It's not even seen in stage 2s, and for sure it's not being seen in advanced stages. So there's something going on, and so it's pure speculation whether it's related to the fact that people are more worried about pancreatic cysts. Our surveillance programs in high-risk individuals account for a certain population, but I'm not sure they account for all that population to switch it. So there's something going on that's changing, but it's a unique facet of stage 1As rather than the global numbers in pancreatic cancer.
And then with that year period of stage 1 progressing to metastatic, that's probably explaining in 2030 being the second cancer mortality related to pancreatic cancer, correct?
I think that's true. And I think also because there have been some significant improvements in other cancers, right? So you're seeing, there have been slow improvements, and there have been some improvements on the therapeutic side for pancreatic cancer, but, we really need to make a real dent for the early detection to really kind of begin to move that needle a bit better.
And then the last question from Rosa, is there also an increase of incidence in patients with young onset? And I believe you just said that in answering David's question.
Yeah, the age of the diagnosis for the stage 1As is going down, not for the other cancers, but for the other stages, but the stage 1A age of diagnosis has gone down. There are data about the overall decrease in the age of diagnosis of pancreatic cancer, and it may in fact be associated with slightly different types of genotypes and molecular profiling. And some of that data has been kind of presented here at Yale in the past. So certainly focusing in on a younger population, hearing about a younger population with pancreatic cancer, which is certainly very concerning.
Well, thank you so much. It'd be great to continue our work here with a focus on Connecticut and how we can really improve upon the lower the incidence and or mortality. So thank you so much.
Thank you. Thanks very much.
Okay, if you can stop sharing. And I'm now delighted to introduce Dr. Vasili Vasiliou, who is chair of our Department of Environmental Health Sciences in the Yale School of Public Health. He is also the Susan Dwight Bliss Professor of Epidemiology and of Ophthalmology and Visual Science and of the Environment. He received his bachelor's in chemistry and PhD in biochemical pharmacology from the University of Ioannina in Greece, and then he trained in gene-environment interactions, molecular toxicology, and pharmacogenetics at the Department of Environmental Health in the College of Medicine at University of Cincinnati. He's established an internationally recognized research program that's been continuously funded by NIH since 1997. And his research interests include the etiology and molecular mechanisms of environmentally induced human disease, such as liver disease, obesity and diabetes, cancer, and neurodegenerative diseases. His laboratory uses state-of-the-art integrated approaches that include metabolomics, lipidomics, exposomics, tissue imaging, mass spec, deep learning, as well as human cohorts and genetically engineered mouse models. And we are delighted to have him here today to talk specifically about alcohol and cancer. Thank you, Vasilis.
Thank you, Melinda. Is my microphone? Yes, my microphone is on. Thank you very much for your nice work. And thanks also, James, for pointing out our T32, who has helped on the studies before. Before I start mine, I also want to address a little bit of the question of Melinda, your question regarding the increased incidence of cancer lately. One of the things that I would like to point out is we have some increases in environmental exposures, especially for the perfluorinated compounds, namely PFAS, which may have contributed in all kinds of tumors. I wish we should have a roundtable another time to discuss about all these factors. So today, what I'm going to talk to you about it is molecular mechanisms of alcohol and cancer. And so we have edited 3 books on alcohol and cancer, and one special issue on chemical-biological interactions, which was based on my 3rd international conference on alcohol and cancer, which we held it here in Newport just before the COVID. As a matter of fact, we just received a note from Springer that our book's doing so good on alcohol and cancer, and they want us to write another one very much, but we will see. So I have, there is this association between alcohol and cancer, and it's actually, you know, there is epidemiological evidence that is convincing that alcohol is associated with several cancers listed in here: mouth, pharynx, larynx, esophagus, colon, and breast for women. And, you know, there is some more breast and liver based on IARC, and there is a probable liver and colorectal for women. One of the things that I have not included in this presentation, though, and I have to say that as well, they have been epidemiological studies which indicates that alcohol sometimes is a positive factor in preventing alcohol. So I don't have time to go with that. I'm going to stick with the mechanisms today, molecular mechanisms of the alcohol-induced cancer. So before we go to that, and since we're talking about epidemiology, I want to tell you the categories of association between alcohol consumption and cancer. And there is first case is sufficient evidence of a causal association, which is the case for every study. There is sufficient evidence of an association. There is limited and suggestive evidence of an association, inadequate or insufficient to determine whether an association exists, limited and suggestive evidence of no association. This has been described very well in a paper by our junior faculty, Josh Wallach. It was in 2020 in the International Journal of Epidemiology, and this is worth reading it. One of the problems with alcohol-based epidemiological studies is most of the studies are based on a questionnaire rather than having clinical data in terms of the alcohol consumption. And we know there are certain biases that could really interfere with the outcome of the studies. And this is most of the time people really are, don't tell the truth. So answering the question, how many drinks you have per week? Well, maybe one, maybe, and could be just a bottle of hard liquor per week. So this may create some problems, and this is what I'm trying to introduce a new term, which is called the alkosome, which is a panel of markers that could indicate what is your alcohol consumption throughout the years in your life. So in December 2020, I was involved in an NCI workshop that we presented, the existing knowledge and evidence and gaps across the cancer continuum regarding the alcohol and cancer. So we have looked at from epidemiology and biology of the alcohol and cancer risk, what needs to be addressed and how we can really work in preventing, this alcohol consumption that could lead to cancer. Again, this is readily available to everybody, and I'm gonna go directly to the mechanism. So I've been working with alcohol metabolism since day one of my graduate studies, and that was the metabolism of alcohol, which actually is involved, is been considered as the mechanism of inducing cancer. So your alcohol, your ethanol, is metabolized by alcohol dehydrogenases to an aldehyde, which is a powerful, it's an aldehyde, it's an electrophile, which is capable of interacting with DNA and protein and forming adducts. And this is a very important molecule in here. So ethanol can also be metabolized by catalase and also by cytochrome P450s, mostly cytochrome P452E1 and to a lesser extent by 1A2. What happens during the ethanol metabolism of acetaldehyde by P450s, you have generation of reactive oxygen species, you have glutathione depletion, which leads to oxidative stress. And as you will see later on, we have the formation of further aldehydes, namely 4-hydroxynonenal and malone dialdehyde, which are further capable of causing DNA and protein adducts. So ALDH2 is the major enzyme which metabolizes acetaldehyde, the major product of ethanol, and it is converted to acetate. And then from the acetate can go to acetyl coenzyme A, which can be used as a building biomolecule for a lot of cells, including the cancer cell. That's another story. We don't have the time to go over that today. So as you can see, the aldehydes and the reactive oxygen species are very important. My lab is focusing in all aldehyde metabolism. I'll show you a little bit more, but we're also looking at ALDH2 and ALDH1B1 and ALDH1A1. This is aldehyde dehydrogenase enzymes, which they take acetaldehyde and convert it to acetate. So again, you can see for the risk factors in terms of alcohol drinking, as mentioned earlier, there is age, the personal history, the family history, race, and also diet, physical activity, obesity, smoking, and alcohol use, as Melinda and James were mentioning above. So what we're doing in my lab is, and I have a center funded by the NIAAA, is we have all you can see here is the ethanol metabolism and also glutathione metabolism, the synthesis of your major antioxidant of your system here. And what you can see on those red letters are the enzymes involved in these pathways. And we have single and double knockouts of all these enzymes, which are involved in the ethanol metabolism and the interplay, and essentially trying to clear up the reactive oxygen species generated and protecting your cells. So we just published a review in Cancers on molecular mechanisms of alcohol-induced colorectal cancer. So this is one of the areas that my lab has been focusing for a number of years. We had the UO1, which I'm planning to go for the renewal as well. And essentially, again, you can see that ethanol metabolism is involved in here. And we have what I described you before, the pathways here. But one of the things I want to tell you is the ethanol per se is not only metabolized by cytochrome P452E1, but constantly increases the levels of cytochrome P452E1. Why this is important? Because 2E1, it can metabolize the procarcinogens into the carcinogens, and that is very important. Another area of the cytochrome the 2E1, it can increase the cell proliferation. And also, as we discussed, it can create reactive oxygen species. The other is the important roles of both cytochrome P452E1 and also, the aldehyde dehydrogenases in retinoic acid homeostasis, which they've been also involved in the whole process. Another important issue here is the ratio between NADH and NAD, which is completely changed based on the ethanol metabolism. Another area which ethanol could affect is the 1-carbon metabolism, and that has effects on the DNA methylation, which can really give a lot of changes in DNA methylation associated with cancer. As I told you before, acetyl coenzyme A can be used as a micromolecule biosynthesis, including in the cancer cells. We have a lot. I don't have the time to go. I just want to give you a brief overview of what's going on. So one of the major things, so here, if we can focus, is you have the adduct formation, the DNA and the proteins. I'm just going to show you some examples of the DNA. I'm going to talk about some the inflammation as well, and we're going to go from there. So what happened with the acetaldehyde and also the other aldehyde generating the DNA adducts? So these aldehydes are potent electrophiles. They can interact with bases of the DNA and they can form adducts. And these adducts, then they are responsible for causing mutations into your transcripts. And acetaldehyde, by also interact with various amines, can give a crotonaldehyde, and this also can form further adducts in here. At the same time, as I mentioned before, you also have an increased lipid peroxidation from acetaldehyde, which is due to the glutathione depletion, which generates further aldehydes, 4-hydroxynonenal and so on. So you have another type of diethanone, desoxyguanosine here, another type of adduct. So it is very well documented that both acetaldehyde per se or the byproduct aldehydes that can interact with DNA, they can cause adducts, and these adducts can lead to mutations and they can cause cancer. This is a causative effect, and this is what helped IARC to establish and categorize as ethanol as a cancer agent. Another area, I am not going to go on these aspects in here. What I want to tell you is that ethanol also can induce inflammation and changes into the cytokines and chemokines, and then they can have various effects into all the signaling, which essentially they can affect the DNA repair mechanisms into the system. So the importance, this is less studied, but this is an area that we should emphasize. And as a matter of fact, this is what I'm going to tell you, what we're planning to do in here. But, again, it is a pathway that ethanol could affect all these pathways in here, including the DNA damage and, and essentially having the more further mutations. So one of the areas that I am planning to expand is the immunometabolism. And this is the interplay between the immunological and metabolic processes. And as a matter of fact, we have published several papers on the role of glutathione in the control of the T cell and macrophage functions. But essentially, again, you can see the mitochondrial metabolism here and how the ethanol metabolism can interfere with that in terms of the cancer incidence. As I told you before, the ethanol can also, alcohol can also affect the 1-carbon metabolism, and ethanol can block the absorption of the folic acid and can interfere with all the folate cycle and the methionine cycle, which eventually, and as you can see here in various stages, through the either interacting with proteins and, forming adducts, or interacting with pathways or absorption of stuff, essentially we have a DNA hypomethylation, which can be associated with cancer. And of course, that's the other pathway that, by interfering with the 1-carbon metabolism, you can have decreased glutathione, which really, gives rise to oxidative stress, which is a result of reactive oxygen species generated by the cytochrome P450 metabolism of the P42. And taking a break here, what I wanted to tell you is, in the slide before here, is the procarcinogens. We have a lot of procarcinogens exposed in our lives. So if alcohol induces the cytochrome P450 2E1, then we have increased levels of the 2E1. So being exposed to dimethyl nitrosamine, for example, or the nitrosamines or the PFAS or everything, then you can have a further activation of these carcinogens, and you can have this generation of reactive oxygen species and further mutation. I'm going to end up very quick. I'm going to talk. I'm not going to spend my time. You all know the incidence of the colon cancer and what is going on, but my lab has been focusing on the colon cancer and alcohol metabolism. We were the first to clone the secondary enzyme. If you remember, acetaldehyde is metabolized to acetate via the aldehyde dehydrogenases. ALDH2 is the major enzyme, but we did found, we cloned this enzyme, and mitochondrial enzyme, which is very similar to ALDH2, and we cloned it, we expressed it throughout the years. We made beautiful antibodies. We made the cover pages of several journals. And we found out, first of all, that metabolizes acetaldehyde with very high affinity, which makes it as equal as ALDH2 in terms of clearing the acetaldehyde. But also what we found out is this enzyme is also involved into the retinoic acid metabolism, which is very important. So one of the things we found out that ALDH1B1 is a potential biomarker for colon cancer. So we have screened several panels of tumors, and we found that especially for colon cancer, every single colon cancer sample that we have tested, it really expresses ALDH1B1 at very high level, and which we didn't see it in lung, breast, on ovary, or anything. And it was especially this. It was not ALDH1A1, it was ALDH1B1. And this has to do also, with the colony formation and everything else. So we also, in addition to colon cancer, we have also found out that ALDH1B1 plays a really big role in pancreatic cancer, trying to make a connection with James' talk earlier on. So this is the ALDH1B1, and you can see there is really high expression of this into the pancreatic adenocarcinoma. So one of the things that actually you can use that as a prognostic marker is you can take the tumor, then you can really isolate ALDH positive and ALDH negative cells, and then you can put them in culture. As you can see, if aldehyde dehydrogenase is not present, you have very small formation of the colonies. But if the ALDH positive, you really have seen the tumor formation. So for somehow, this ALDH is involved in the tumor proliferation, initiation. I don't know. We're still looking at the role of this throughout, either through the DNA repair mechanisms or through retinoic acid. It's an active area. So we have several papers on the how the ALDH1B1 could be that, and we found out that can be modulating the Wnt beta-catenin pathway and the also P1K, the AKT signaling pathway. But the most important thing, and this is a work that we have done, and we have identified that it is immunohistochemical marker for colorectal cancers, and we look at that on several samples, and including some comparisons that we have done here at Yale. So it's a very strong marker for that, and as I said, we're still looking on what is going on. So one of the way that we have trying to address the role of this is we have generated a specific knockout. So it's a tissue-specific inducible knockout on a triple knockout mice. So the APC mean mice, we know that they are forming many cancers. So anyway, to make time is running, we have generated a triple knockout of this with APC and everything. So these mice, we have found out that this is the APC-ALH knockout, and this is the wild-type knockout mice. And this is the experiment. This is how we generated it. It was a painful process. A triple knockout is not an easy way to do it. But the way that you're doing that is you have an inducible model. You treat them with tamoxifen, you delete the gene, and then you can further figure out of what's going on. So we did not find any difference in the vehicle-treated mice, and also in the tamoxifen-treated groups, adenomas were not evident in jejunum or ileum on these mice. However, we did find the colorectal adenomas, which in the knockout developed significant low numbers of the macroadenomas, and there was a significant reduction in the total macroadenomas in the knockout. So when we knock out the gene in a model that induces colorectal adenomas, it is the gene, if you knock out the aldehyde dehydrogenase, you can see that you reduce. So this confirms our initial studies that the ALDH1B1, it is involved in the development of adenocellular carcinomas. And we have been trying to dissect more involvement of this gene, and we didn't see any difference at the P53 level, but we did see a decreased expression of the beta-catenin in the knockout ALDH beta-catenin ALDH knockout for beta-catenin expression, which you know that beta-catenin plays an important role in the development of colon carcinoma. So the expression of ALDH1B1 could be used as a novel biomarker in identified colorectal cancers and also in pancreatic cancers. The ALDH knockdown caused a dramatic reduction of tumor growth and loss of function of APC in absence of ALDH1B1, reduces the colon adenoma, and in turn delays the tumorigenesis and the progression of cancer. In that, to that effect, I have collaborated with NCATs, and we have developed specific inhibitors of the aldehyde dehydrogenases that could be used for delaying the progression of the tumor once it's been detected. So looks like the ALDH1B1 is a key player in this carcinogenesis. So if I can go back a little bit on the molecular mechanisms to conclude my studies in here, and the directions is the alcohol metabolism is a key player through the cytochrome P450 or through the aldehydes generated in here, and then you have also the changes in the NADH ratio, which we can also have epigenetic modifications. Remember, reactive oxygen species, they can cause the inflammation, they can lead to cancer. In addition to that, you also, as mentioned, this is very important here. This is especially with increased exposure to environmental carcinogens. The high levels of the 2E1, they can lead to DNA damage and mutations, and they can cause cancer. And of course, you know, the DNA repair can also be affected by both the 2E1 and also the alcohol metabolism. So a very important process in here, and this is what we're focusing as well, is how ethanol metabolism could have defects in the DNA repair as well. And as I mentioned also, you know, we have the 1-carbon metabolism and DNA methylation. I didn't have time to go on your microRNA in here, but also I did mention the crosstalk between these pathways, the alcohol and the immune system, which eventually they can end up in causing epigenetic modifications that they can cause to cancer. This is a slide the best slide that I've seen in terms of concluding all the effects of the alcohol in terms of the cancer. And this was from the former director of the metabolism of the NIAAA, Sam Zakari, who is a very good friend and colleague of mine. And I want to tell you, this slide is, I'm, you know, a little bit sad, but the 5th International Conference on Alcohol and Cancer was going to be in Greece 2021. Unfortunately, with the COVID, we have to move it ahead. And probably it's not going to be 2022, it's going to be probably 2023. And I expect to see quite a few of our Yale colleagues here. Thank you, Melinda. I'm going to stop and get some time for questions.
Thank you, Vasilis. Count me in on a trip to Greece. I know you just returned and I wasn't invited. Anyway, to the topic at hand. I was intrigued at the beginning of your talk when you were talking about measurement error and the fact that often we measure alcohol intake through questionnaire, and we know there's a lot of misclassification with that, underreporting perhaps.
Yes. Yes.
And then that misclassification really attenuates the hazard ratio to the null. So in reality, the impact of alcohol on cancer is probably even higher than noted in studies. But you then mentioned what you call an alcoholsome or something of a panel of markers. Can you discuss that briefly a little bit more?
Absolutely. So you see, alcohosome is a term similar to the exposome, and I'm trying to pattern actually this word. So I'm trying to figure out through animal studies and human studies how I can define a motif of changes that you can utilize by looking at the blood and finally modeling it to how much alcohol one person has consumed during lifetime. This is not an easy task, okay? So, because one of the things is, remember, you can drink alcohol and then even if you measure a few of the enzymes, your hepatic enzymes, if you have abstinence of alcohol, the liver recovers. So you don't know to how much alcohol you have drunk. So what we're trying to do is we're trying to have a more educated measurement of determining how much alcohol a person have done throughout life. And the same thing as the exposome concept. As I said, it's not easy, but we're trying to do that, and we're trying to do with animal studies and also with human studies. This is one of the ways that it can help really the epidemiology to assess, you know, the causal effects of alcohol, because this is another area of big debate. Melinda, as you know, there was a couple of years ago, there was one paper from New Zealand and it's saying, oh, the women, even if they have one drink per year, they can develop breast cancer. And if you look at really at the studies, they were, based everything on just the questionnaires. And as I said, this is not the accurate measure to do this.
Yeah, yes, I agree. So much work to be done. Well, thank you so much to both of our speakers today. And if others who tuned in have questions, I'm sure you can reach out to both our speakers via email, and hopefully there will be some collaborations initiated from today's talk. Thank you both. Have a great rest of the day.
Thank you.